Hey guys, so in this video we are going to start the beginning of chapter 14 and talk about DNA and all the different scientists who led us to where we are today. So in the beginning we're going to talk about chromosomes, which we have mentioned before when we talked about mitosis and meiosis, but that DNA is going to be very long strands of DNA wrapped around histone proteins, but then coiled together. They are tightly coiled together in the form of chromosomes. We have 23 pairs of chromosomes for a total of 46 chromosomes in every somatic cell. So every cell in the normal cell in your body is going to have 46 chromosomes, 23 pairs of chromosomes. If you're talking about reproductive cells like gametes, those are going to just have 23 chromosomes total. So those are the chromosomal genome. But there's also the extra chromosomal genome like things in the mitochondria. As we mentioned before when we talked about the chapter with cell parts, that some cell parts have their own genome, and mitochondria were one of those, and chloroplasts were one of those, and those led us to the symbiotic theory that the chloroplasts and mitochondria were originally prokaryotes, and that's why they have their own genome. And there was some other evidence for that too, but that was one of them, that they had their own genome. So there is genetic material outside of just the chromosomes. When we have cells that are going to produce their gametes, we talk about that part of leading up to mitosis and meiosis, was the cell is going to get bigger and reproduce what organelles it has inside of itself. So when we have gametes, the mitochondria that's coming from parent to offspring comes from the mother. Mom's mitochondria in her egg cells gets passed down. Dad's nuclear material in his sperm gets passed down, but not much else from the sperm actually comes into the new potential zygote. It's the nuclear material. So we have chromosomal material, DNA found inside of our eukaryotic nuclei, our membrane-bound nuclei. But there's also mitochondrial genome found in the mitochondria, and that will be in mother's egg cell when the stem cell that creates the eggs undergoes meiosis and produces an egg at the end. That's the mitochondrial genome that passes from mom down to mother's offspring. Genes are going to be the functional units. They are going to code for a product, for some sort of trait, and we have lots of genes. In the textbook says, according to the textbook, that we have 3 billion base pairs, and we're going to talk about base pairs and what that means. 3 billion base pairs and 20,000, over 20,000 functional genes in our cells. We start off with Dolly the sheep here, and I remember this because I am old, but I remember when Dolly was cloned. This was in '96, and so what they did was they took the mammary cell, hence the name Dolly. They took a mammary cell, a mammary stem cell, out of one sheep, and that stem cell had both sets of chromosomes because it was a somatic cell, so it had mom's and dad's set. There was 23 pairs of chromosomes in this mammary stem cell. And they took this mammary stem cell, took the nuclear material from that stem cell and put it into a donor egg. And then they did some sort of process to that donor egg, and I don't know all the specifics on it, but they got that donor egg and stimulated it so it started to act like it was already a fertilized zygote. And in theory, it was. It had a diploid nucleus inside this egg. So once they started it going and undergoing cleavage, that was reproductive cloning, and the first one was Dolly the sheep. So how do we even know about DNA? How are we sure that it was DNA inside the cells? So we have to go back in time to figure out who were some of these first scientists. And so some of the very first scientists discovering DNA, talking about DNA, was Michener. So Michener was around in the 1800s, and about 1860, he was playing around trying to figure out what was inside the cells. And so we have an animation here. Again, on all these links, if you want them to work, put your PowerPoint that you've downloaded to your device, put it in the slideshow mode, and you can click on all these links. So if you click on this link, you can see the animation that's talking about Michener. And he wanted to figure out what's inside the cells. So he was a doctor and had ready access to materials from other people. So what he did, it's kind of gross, but he took these bandages out of the hospital, and he took these bandages that were being, when somebody has a wound and they clean the dressing, they take off the old dressing and they put on new fresh stuff. So he took buckets of old pussy bandages and put them in solution and lysed the cells. So now we pulled the nucleus out of the cell. And then he put in an alkaline solution on those nuclei and ruptured the nuclear membrane. And this stuff came out of it. So there was stuff inside the nucleus. And so he called this stuff nuclein. Nuclein. So Meissner gets the credit for calling this stuff nuclein because it's inside the nucleus. We had some other scientists come along after that. Altman was after that, and he discovered that that material that was inside the nuclein, inside the nucleus, had acidic properties. So now we call them nucleic acids. And there was another scientist that came along after that. Cosell was studying the nucleic acids and discovered that there's 5 different nucleotide bases. So the nucleotide bases, we divide them into 2 groups. There are pyrimidines and there are purines. And pyrimidines are best buddies with pyrimidines. They are best buddies. They're going to bond with hydrogen bonds and give us those base pairs. So adenine is always going to, when we're talking about DNA, bind with thymine. And guanine always binds with cytosine. C's and G's always go together. A's and T's always go together. And we're going to talk about Chargaff's rule in just a little bit. The difference that there is a 5th nitrogenous base, and that is with RNA. So RNA is not going to use thymine. RNA is going to use uracil. So we have Cosell to thank for discovering the 5 different nitrogenous bases, 3 pyrimidines and 2 purines. And these are the beginning of discovery of DNA. So now we know that there's this molecule and it's inside the nucleus, inside of all the cells. Let's go on to another scientist. So Griffith is another scientist, and this experiment is huge, and every biology class always talks about this. So with Griffith's experiment, there's strep pneumoniae. There's a bacteria, strep pneumoniae, and there's 2 strains of it. There's a good one and a bad one. The rough strain has R for rough, and this bacteria does not have a sugary coating, a capsule on the outside. So he's non-pathogenic, meaning he's not going to get you sick. Then this other version, the S strain of strep pneumoniae, has a capsule on the outside. So they look smooth when you look at them under the microscope. And this smooth strain, this capsule, helps it attach onto the cells and invade. So that's what makes it more pathogenic than the rough one who didn't have the capsule. So 2 strains, rough and smooth. And so when he took the rough strain and Griffith injected the rough strain into the mouse, he survived. The mouse survived. So rough strain, non-pathogenic, totally fine. What about the S strain? We know if we put the S strain in and inject it into the mouse, that mouse is going to die. What if we treated the S strain like we do our food prep that we know of now, that we cook it? So if we take this bacteria and we heat it up a lot, we will kill it. So let's do heat-killed smooth strain and inject that into a mouse. Well, if we inject the heat-killed one, it was okay. The mouse survived. Okay, awesome. So if we kill the S strain, it lives. If we use the rough strain, it lives. Let's combine the 2 together and see what happens. So with the rough strain, which is the good guy, and we combine it with heat-killed, the dead S strain, we put those 2 together in the same solution, the same media, and we inject that into the mouse died. Why is that? Well, it turns out it is because of transduction. So bacteria can find genomic material out there, like from the smooth strain, because we killed it and it's in the solution. Bacteria can take that DNA into their cells, and they'll put it in a single strand DNA and incorporate it in. And then they'll put that in their chromosomes and they'll use it. So they'll pull in this DNA, a single strand DNA. They will make another copy of it. So now it's double stranded DNA, and they put that into their chromosomes and they will use it. They will express it. So when experiment number 2 happened, we took a good bacteria, paired up with a dead bad bacteria, and we made the good guy into a bad guy because of transduction. This is horizontal gene transfer. We knew that genes transferred from parents to offspring, but we did not know that genes transferred from a friend to another friend. This was groundbreaking news. So this is Griffith's transformational experiment, horizontal gene transfer. So we took the good guy, the rough guy, paired up with a dead bad guy, and when we put those two together and we injected it in the mouse had passed away. In the next experiment, we're going to take that strain recovered from the dead mouse and see if we're going to perform postpostulate. We're going to pull it from the dead mouse, isolate it, and put it into another mouse, and that mouse dies. So that confirms that it is legitimately the good guy has turned into a bad guy, and we are now pathogentic because of this. So horizontal gene transfer was discovered by Griffith. He called it the transformation principle. It's horizontal gene transfer, going from this bacteria to this bacteria instead of parent to offspring. Horizontal transformation experiment with Griffith. Avery, McLeod, and McCarty was like, I don't know. I think you're full of it. What if it's not the DNA? What if that bacteria picked up the protein from the other bacteria? And maybe it's the protein. Or maybe it's the RNA. How do you know that it's the DNA that made that other strain kick over and become pathogenic? That's why I like science, because there's always somebody out there to test and test and test and prove somebody right or wrong. So Avery, McLeod, and McCarty decided to perform a test. And so here we have our control. In the control, we don't have any enzymes. It's just the control. So we have all the proteins, all the RNA, all of the DNA in here as these different colored molecules. Proteins, DNA, and RNA are all present in here. And when we expose all of these from the heat-killed strain, the protein, DNA, and RNA from the S strain, and we put it around the R cells, we are now transforming those rough cells into bad guys using the smooth cells stuff. What specific stuff is it? Because we had all three in here. Was it the proteins, was it the DNA, or was it the RNA? That's what we're going to figure out. So in this next one, we are going to use a protease. A protease is an enzyme that breaks down the proteins. So now we're going to eliminate the proteins, and we're going to see if it was the proteins, because if it was the proteins that did it and we broke down the proteins, we shouldn't see the cells transform, but we end up seeing the cells transform from good cells to bad cells. So it's not the proteins, because even though we eliminated the proteins, the cells still transformed. Maybe it's the RNA. Let's eliminate the RNA. When we eliminate the RNA with a ribonuclease, and that's going to bust up the RNA. And now we are left with the DNA and the proteins, and the DNA and the proteins were able to transform the R cells into modified pathogenic cells because of the S. So this one still didn't work. So we know it's not proteins and it's not the RNA. Let's try the DNA. So with DNA, we are going to use deoxyribonucleases, and they degrade DNA. They're going to bust up the DNA, and that's going to leave the proteins and the RNA left in solution. When we leave the proteins and the RNA left in solution and we expose the R cells, nothing happened. Those R cells are still non-pathogenic. They did not turn over from good guys to bad guys. So the S cells are absent, it has to be the DNA. So it wasn't proteins and it wasn't RNA, it was DNA. That was the source of transformation that was getting transferred from the good bacteria, from the bad bacteria that was heat killed into the good bacteria via transduction. So Avery, McLeod, and McCarty reinforced Griffith's experiment with the transformation principle being the molecule of DNA and not RNA and not proteins. Another experiment. This is Chase and Hershey, and they too were like, nah, I think you're fooling it. I think there's something wrong with your procedure, and I'm going to do my own thing, and I'm going to test it and see what it is. So with this one, they used a bacteriophage, and bacteriophages are viruses that infect bacteria. So they use a bacteriophage, and it infects E. coli. So we use E. coli all the time in the lab. Let's see exactly what it is with this experiment. So we're going to tag it. We're going to tag the DNA is going to be labeled here with a fluorescent phosphorus label, because every nucleotide is going to have sugary backbone, a phosphorus, and a nitrogenous base, a pyrimidine or a purine. So if we label the phosphorus, we know we're just labeling the DNA. So here we have tagged with a label the DNA found inside this bacteriophage. Every virus has a genome inside, whether that's DNA or RNA. In this case, this bacteriophage has DNA. Every virus also has a protein coat on the outside, this capsid of this protein coat. So with the protein coat, we've labeled that. So now we have sulfur labeled protein coat, and here we have the phosphorus labeled DNA. So Chase and Hershey are trying to figure out what gets passed on. Is it protein or is it DNA? So they have a bacteriophage. It infects the E. coli cells. There's our little E. coli cells. And we know that bacteriophage is going to inject the genome into the E. coli cell, and then that genome hijacks the cell. But they weren't totally sure exactly what it is that hijacks the cell. Is it the genome or is it the protein? So they took the cells, they exposed them to either the P-labeled DNA bacteriophage, or they exposed them to the S-labeled protein-coded flag bacteriophage. And then they are going to put those cells that are growing in the media in solution, and they're going to spin them down. And they're going to put them in a centrifuge. And if anybody's been, had the experience of being in, I don't know, one of those fair rides that spins around, if you've been to Six Flags and you ever sit in those swings and then they spin the swings around, the heavy stuff flies to the outside on a centrifuge. So the heavy stuff ends up at the bottom, and the lighter stuff stays in closer on the centrifuge. So the lightest stuff will be up here, and the heaviest stuff's at the bottom. Our cell sizes, eukaryotic cells are in general really big. Prokaryotic cells are usually much smaller. Viruses are even smaller than that. So viruses are going to be at the top of the tube, and the prokaryotic E. coli cells are going to be at the bottom of the tube. And when we spin this down, we're going to take this out and we'll harvest the cells. So what actually got transferred into those E. coli cells? So here we could see, oh, there's those flagged genomes that we put originally in that bacteriophage, and they have transferred into the E. coli. On our protein ones, there's nothing here. Where did it go? Oh, that's right. It's still hanging out in here. So it's not the protein that gets transferred into the cells. The protein from the bacteriophage on the outside of the virus, it does not go into the E. coli cells. It's the DNA that goes into the E. coli cells. So Chase and Hershey proved that the genetic material, the DNA, is what is involved in inheritance, and that is what gets passed down and transformed things, whether it's vertical transfer from parent to offspring, or it's horizontal transfer via transduction, or conjugation, or all of that stuff is genetic material. That genetic material is DNA. So this is Chase and Hershey's experiment proving that it's not the proteins, it's the DNA. The last thing in this section is Chargaff's rule. with Chargaff's rule, he was analyzing those nucleotides, those nitrogenous bases, and seeing that he was looking at lots of different species. And if he looks within one species, he was noticing that the ratio of adenine is the same as thymine. And if we look at the guanine and the cytosine, they have the same ratio too. So if we had, I'm just making this stuff up, if we had 10 guanines, we're going to find that we have 10 cytosines. And if we have 15 adenines, we're going to have 15 thymines. So that ratio, A's and T's go together and C's and G's go together. That's called Chargaff's rule. We know this. In every species, you're going to find this ratio. It's not going to be the same if you're looking at one species versus another species. That ratio is not going to be the same. But within the same species, A's and T's are going to bond together, and C's and G's are going to bond together. This is Chargaff's rule. So that finishes the first section, and we'll start again on another video with the second section.